Welcome to this comprehensive lecture on the life cycle of Plasmodium vivax. Today, we are going to explore one of nature's most fascinating and deadly biological spies. We will understand exactly how it survives, how it multiplies exponentially, and how it spreads malaria across populations. By the end of this session, you will see how biology and mathematics come together to help us fight this disease. To start, imagine a master spy who must use two completely different disguises in two different countries to complete a highly complex mission. If they only visit one country, the mission fails completely and they cannot survive. This is exactly how the parasite that causes malaria, known as Plasmodium vivax, operates in the real world. It cannot survive by just living inside a human, nor can it survive by just living inside a mosquito. It absolutely needs both environments to complete its life journey. When a mosquito bites you, it might be dropping off these tiny biological spies into your bloodstream. Now that we understand its double life intuitively, let us look at the formal biological definition. Plasmodium vivax has what we call a digenetic life cycle. The scientific term digenetic simply means that the organism requires two distinct hosts to complete its full life cycle. The primary, or definitive host, is the female Anopheles mosquito. This is where the parasite undergoes its sexual reproduction phase. On the other hand, the secondary, or intermediate host, is the human being. Inside the human, the parasite undergoes massive asexual multiplication to rapidly build up its numbers before moving on. Building on that, let us zoom in on what exactly happens inside the human body. The human phase of the life cycle is further divided into two main parts. First is the hepatic, or liver phase. Here, the parasites hide from the immune system and begin to multiply initially. Next is the erythrocytic, or red blood cell phase. This is the most destructive part of the cycle. It is this red blood cell phase that causes the classic malaria symptoms of cyclical fevers, chills, and exhaustion, as hundreds of thousands of cells burst simultaneously. While the parasite wreaks havoc in the human bloodstream, its journey continues only if another mosquito bites the infected person. Inside the mosquito, the parasite shifts its strategy entirely. It undergoes a process called gametogony to form sex cells, which then fuse. This is followed by sporogony to create infectious spores. These newly formed spores eventually migrate all the way to the mosquito's salivary glands. They sit there, perfectly positioned and waiting, ready to be injected into the next human host the moment the mosquito takes its next blood meal. Understanding these specific stages is not just an academic exercise. It is absolutely critical for global public health and modern medicine. Because we know about the dormant liver stage, known as hypnozoites, we can use targeted antimalarial drugs like primaquine to seek out and kill them. Furthermore, knowing that the mosquito is the definitive host allows us to implement powerful vector control strategies. By using tools like insecticide-treated bed nets and indoor spraying, we can break the transmission cycle entirely and protect vulnerable populations from infection. This biological multiplication leads us to an important mathematical model. When the parasite enters the red blood cells, it does not just add to its numbers. It multiplies exponentially through a process called schizogony. The formula Pn equals P0 times m to the power of n describes this explosive growth. Pn represents the total number of parasites present after a specific time. P0 is the initial number of parasites that entered the blood. M stands for the multiplication factor, which is typically 16 for this species. Finally, N represents the number of 48-hour reproductive cycles. As N increases, the total parasite load explodes exponentially. Before we move on, let us see this formula in action with a concrete numerical example. Suppose a patient starts with a small initial population of 100 parasites. We know the multiplication factor is 16, and we want to find the total parasite load after 3 reproductive cycles. We plug these known values into our exponential growth formula. First, we calculate 16 raised to the power of 3, which gives us 4,096. Multiplying this by our initial 100 parasites gives us a massive final count of 409,600. This physically explains why symptoms escalate so rapidly. Now, let us look at a slightly more complex, real-world application involving symptom thresholds. A patient typically experiences severe malaria fevers when their total parasite count reaches 2,560,000. If the initial infection was 10,000 parasites, how long does it take to reach this dangerous threshold? We set up our equation with the known values, placing the threshold on the left and the starting conditions on the right. The final answer turns out to be 2 cycles, which equals 4 days. But how exactly do we isolate the exponent to find this result? Let us break down the mathematical derivation next. This brings us to the step-by-step derivation of our infection timeline. We start with our populated exponential equation. Our goal here is to work backward and solve for the unknown exponent n, which represents the number of reproductive cycles. The first algebraic step is to isolate the term containing the exponent. We do this by dividing both sides of the equation by the initial population of 10,000. This mathematically removes the starting condition from the right side, simplifying the large numbers beautifully down to 256 equals 16 to the power of n. Continuing from our simplified equation, we now need to find what power we must raise 16 to in order to get 256. By simple multiplication, we know that 16 multiplied by 16 equals 256. Therefore, the exponent n must be exactly equal to 2. This means it takes exactly 2 reproductive cycles to reach the fever-inducing threshold. Because each erythrocytic cycle takes exactly 48 hours, or 2 days, we multiply the 2 cycles by 2 days per cycle to get a final timeline of exactly 4 days until severe symptoms appear. Now that we have proven the mathematical timeline, let us visualize the entire biological process. On the screen is a detailed schematic of the Plasmodium vivax life cycle. Notice how it is split perfectly down the middle. On the left, in the human host, you can see the parasites multiplying in the liver and then bursting from the red blood cells in a continuous loop. On the right, in the mosquito host, you see the sexual reproduction phase in the mosquito gut, eventually leading to sporozoites in the salivary glands, ready to be injected into the next human. This diagram perfectly captures the dual nature of the parasite. To wrap up our lecture, let us review the most important ideas we have covered today. First, Plasmodium vivax relies on a complex digenetic life cycle, requiring both a human and a mosquito host to survive. Second, it is the explosive, exponential asexual multiplication inside human red blood cells that causes the devastating symptoms of malaria. We can model this mathematically to predict symptom onset. Finally, mastering these biological details empowers us to fight back with targeted drugs and vector control. You now have a complete understanding of this fascinating and dangerous parasite.